Probably 3 gig per second.
Oh, wow. Okay. Where is that Dr. Seuss sponge?
Is it saved at that?
What's that?
Is it all saved at that size? 3
3
gigabytes a second? But we don't store it at that size, do we?
I believe so, yeah. So all that fancy gear is there for. We don't stream it at that, obviously. We need a
minute
much
or a
bigger
second?
satellite.
All right, so the previous dive,
Yeah, per second.
which was dive 1889, we collected, would that be 23.6 gigabytes of data? So that's the video and all the raw feeds and so forth.
Less than I thought.
Dan, you should come up.
Yeah.
That's a great question, though, just to even think about it. I know Sarah over there in the data chair is collecting still images as well. Sometimes many per minute.
Are you saying we don't record it at that?
Each 15-minute clip is about 18 gigabytes of the full resolution video.
Oh, wow.
18?
18 gigabytes for 15
80.
minutes. 18.
Oh, 18.
Yeah.
No.
Love a good crinoid.
It's a Brisingid.
Oh, sea star.
Brisingid?
Closely related.
Getting tentacles out there.
Yeah. Little zoom. So pretty.
Those are, yeah, like almost touching in the middle. He's more curled than I've seen the others.
Okay, let's split.
I think your hardware there has the capability to do 86 channels at 12G. Just an insane amount of...
That's a crinoid up there.
There.
I think
go.
that's a
And
stalked
black
black
coral.
coral?
And then this is a...
I think it's a
is this
stalked crinoid.
Oh, weird.
Oh, but we're
okay.
looking at the one in the back, the yellow
Oh,
one.
interesting.
Oh,
I think that's a stalked crinoid.
Can
we
I
don't
get a
see
zoom?
them very often.
Well, yeah, I think you're right.
I love them.
you love everything today. Usually it's just the rocks, but today you're
Well,
loving
you
on the
can-
fish.
Those
beautiful.
stalked crinoids, you can get fossils of them.
Oh, wow, really?
Yeah.
Do their stalks still have the little discs?
I think these might have the same discs, yeah. But yeah, you can see in the, Sarah's right, in the fossils that
Okay.
we learn
gonna
about
be
in
here.
geology class, you see little discs that are the stalks that make up the stalks. And then you also see in the fossils, they look like flowers, but they're crinoids. Chris has 3 crinoid fossils. That's how nice they are.
With the head too, the flower
Yeah.
part? Wow, that's rare to preserve the top.
Yeah. I think it's in the Devonian, there's places in Pennsylvania where the fossils indicate that there were crinoid fields under the water. You just dozens and dozens of crinoids, like a field of flowers.
Yeah, I think there's some from the Midwest as well.
Yeah.
In the Los Angeles Museum of Natural History.
Do you ever get the fossils at Agate Beach there? And
I haven't got the fossils at Agate Beach. but I would like to go see them.
Oh, Oregon coast.
Yeah. There's a guy you can hire, they call the fossil guy. And
Yeah.
it's amazing. He can walk 10 feet, pick up one and crack it open, and there's a fossil.
No kidding.
Wow.
Oh. I gotta be.
Also hunter for hire.
Yes.
I mean, that sounds like a great Black Coral Friday deal right there. I'd go for
I wonder if
that.
he plants them. He's like, I'm gonna go drop these 5 corals.
He's got like GPS
Yeah.
location.
Oh, the
Never took Andrea for a cynic.
He's like, they want a shark tooth. I'm sure they look the type.
the beach is eroding there, and you can see where they're falling out there.
Oh, I would love to go see that.
I'm going to go out on a stocked crinoid here and say that that was a Bathycrinus species that we saw.
Oh,
but,
really? A Bathycrinus, yeah.
Yeah.
There's another branch coral on the right. I mean, branch
Oh, yeah,
bamboo.
bamboo branch. Yep.
It's funny, we haven't really gotten back into high density
No, we haven't.
We're not far off.
sponges. Yeah, we're like not even 100 meters away
Yeah,
from
which
where we came down.
we still haven't, I don't think, pinpointed the variable.
Which makes it even more amazing that on the first dive, you know, that they planned out, we kind of dropped right into it. You know,
Yeah.
it was.
Well, the ridge is a good spot, but off the ridge, it's pretty hard to predict where it's
Yep.
gonna.
Okay, we're getting into more
Start,
high density
stop.
corals here though.
Yeah. In
Yeah.
front of us, yeah.
Yep,
Yeah.
starting to pick up. But yeah, it was pretty amazing, the dive site that Chris selected.
So we're getting a question here from a viewer. Thanks for joining us. Were these underwater volcanoes ever above water?
This one was, because it's a guyot, it indicates that it was a volcano once above water, and as it subsided, basically wave erosion and coral growth both combined created this flat top. And so when you see a flat top of a seamount, it indicates that it is a guyot and that it was once above the surface. Another thing you can do is go to the guyot face, actually, and if you can see the limestone or carbonate platform that we call it, you know that it was basically keeping pace with the sea surface as the seamount was subsiding. The coral growth was keeping pace with the subsidence, and so keeping the island basically at the surface.
Totally.
Look at that sponge.
I know, I really want to look at this sponge. You don't have to tell me twice. Very
Where the
interested
lasers
in
are?
this. Yeah. Like, what is happening here? What even are you, buddy?
He looks like that sponge that looks like macaroni.
Yeah, it's very macaroni-like.
I forget.
Wow.
Yeah.
Okay, zoom.
Wow.
Oh my gosh.
Very cool. Zoom in more.
Yeah. Whoa.
That's amazing.
That is amazing.
It looks like arteries, which I
Yeah.
guess,
Yeah,
I mean,
heart valves. And
yeah.
That is a beautiful design.
see if I can identify that one.
Okay.
So if the seamount was above water, it will have a flat top when it erodes, and if it wasn't, it will still be pointy?
Exactly. Or ridge-like.
That was a super cool sponge.
Yeah, I don't
Yes,
think
that
I've ever
is
seen one
very
like
cool.
that.
I definitely have not. about my armrests. I love these.
So we have about 20 minutes left on our watch. Please keep those questions coming. You can submit questions at our website, nautiluslive.org. We will be handing it off to the 4 to 8 watch, who I believe will be finishing out this dive with you.
And stay tuned. Do not leave us for Black Friday because we are, as Stephen said, Black Coral Friday, here to entertain you with the deep sea biology.
Ooh, this is a really nice patch here.
Yeah.
Yeah, it's definitely picking it up here, the diversity and density.
Yeah, it is. Yeah, I've seen a sponge before. It wasn't on my dive. I was in the lounge, and it looked like that a little bit. And I said it looked like a macaroni pasta sponge.
Yeah, totally.
Yeah.
I'm with you.
But I don't
like
remember
bunch of elbows glued together.
Exactly, but I cannot remember the name.
Faridae is a guest from the science chat. Faridae. Oops. Let's check it out.
Yep, it could be a Faraday sponge.
Oh yeah, that one looks
Aca erecta. That looks like it.
Yeah.
Faridae.
Faraday.
You guys have an ID on the macaroni sponge?
Yeah, Faraday. It's going to be a family.
Oka.
Faria
corals
genus.
there to the right in Argus.
Oh yeah, totally.
Aca erecta.
Look at
Faridae?
that.
Do we think it's Faridae?
Faraday.
Faraday.
That's beautiful. It's looking upslope.
But ask Chris.
Yeah.
He'll be up soon.
I wonder if he'll know what we're talking about if we say, you know, that elbow macaroni sponge?
Oh yeah.
You
How
think
could
he
you
will?
look at that and think of anything else?
Okay, see, I didn't know if it was just me.
So yeah, Guyot is a very flat-topped extinct volcano. It's not a caldera. It's not forming a depression. It's flat due to erosion and then rebuild by coral reefs. And so the giveaway is when you can see that rock that is not volcanic but is created by the reef, the structure rather.
Calcium carbonate or limestone or, yeah, basically an ancient coral reef.
I just think this is really cute.
That's great.
This is one of those starfish that's been here for years eating the same coral.
Sure.
And I think there's a little squat lobster to the left on the bottom, a white squat lobster.
Oh yeah, the rocks.
Oh yeah.
Wasn't there a certain squat lobster we were looking for yesterday?
With the long arms and
The white
a
one
wide body.
with the
Okay.
stubby arms. Yeah.
Oh, stubby arms, opposite of long.
This
You
one's kind
can
of stubby.
you can zoom, see if you can get a little more on the squat lobster.
Sure.
There he is.
Look at him. He does have short arms.
Yeah.
He does have short arms. Poor
think-
guy.
I think we were
Also, his
also
arms are as long as his body.
I mean,
compared. I mean,
Yeah.
I'll say he has strong arms.
Okay, go wide.
You're going to have to come out ahead there.
Okay, I gotta get cracking. That was a worthwhile stop. more squat lobster. He's already called a squat lobster.
Slide to the east a bit there,
Yeah,
Gabby.
sure.
Wait.
We weren't trying to make fun of him. Somebody was just looking
No.
for one with short arms.
That's right. That's right.
Yeah, actually, he was our dream squat lobster. He had short arms.
Andrea's looking for chunky rocks. Some people are looking for short-armed squat lobsters, you know? We all have our thing. So, Andrea, how long ago would this seamount, the Don Quixote seamount,
that's
have been
beautiful.
above water?
So if it formed between 70 or 90 million years ago, it would be somewhere time around that. So it takes
northeast,
probably
so.
maybe 5 to, depends on the size of the seamount, but
You're downhill.
it can take 5 to 8 maybe million years for it to subside. Ooh, look at that.
Yes, it's beautiful, but I must fly.
Yes, you are flying. I think
Fly
that
like
was
the
another
wind.
stemmed crinoid.
Yeah, I saw that. It was white this time.
Yeah.
Is that another one on the right?
Nope, moving too fast.
Keep on
I shouldn't have
trucking.
let myself get behind.
Looks pretty steep.
It is pretty steep.
Yeah.
Same thing I was thinking.
I was poorly disciplined. I'm really sorry.
And we can't get that far behind on the, otherwise Argus will hit the hill and drag Herc up the hill.
It won't be pretty. It was the short arm squat lobster, though.
It was worth it.
It was worth it. But we did miss the stock crinoid as a result.
Well, our delta gets in the high 20s and our altitude gets in the low teens.
I know, I know.
No choice but to come up. You're back in the box there.
Yeah.
So we
I
got
can look at this.
we got a question
sponge.
here for everybody on the watch. Have you ever been on a dive where you've discovered a skeleton, like a whale skeleton or some other kind of?
Yeah,
Oh, was somebody here on the whale fall a few years ago?
yeah.
That was you, Dan?
Yeah, that's on the website. There's a really nice 3D model of it. So there's one of the branched guys.
Mm-hmm.
Yeah, we were trucking along across kind of a flat, boring spot trying to get to another seamound to look at the octopus garden. And we spotted some anomaly on the sonar and kind of scooted over that way, and it was a pretty epic whale fall. There was tons of the same kind of octopus that we were looking for all over it, and
The moose octopus.
really fat fish. And
We're back in the dense sponges here.
yeah,
Let's drop a target, Nav.
I just can't believe the odds against coming across a whale fall on the seafloor. They're so enormous.
there's one in British Columbia that when we do ONC, they go look at every year. They've been studying it for well over a decade.
Oh, yeah. There's,
Swimming crinoid.
oh, yeah. Oh, it's so weird. That always makes
yeah,
my
right.
skin
Oh,
crawl.
yeah,
I
it's
love
a
it.
swimming
I love it.
crinoid. I love
Yeah.
him too. He's like, no, later.
So cool.
Don't mess with me. I
You
got
can
places
zoom.
to be.
At, oh, I blew it. Sorry. At Palmer Station in Antarctica, there's, you can find a whale fall because there'll be an oily slick on the surface and there'll be seabirds everywhere.
Mm-hmm.
And they just haunt it for seasons. Because it's just delicious, I guess.
There
Yeah, it was one
Can we move 50
very
meters?
smelly ROV when we
There.
brought it
Oh,
up after
God,
getting
I
some
can
samples with the
imagine.
whole boat and the lab, everything. It's just rank for days.
What kind of samples did you guys get?
They picked up a few bones, and I don't know. I didn't look
Bridge
too
now.
closely. We did on the one in BC, they
go
finally decided to pick up another
055
one of the vertebrae.
50 meters.
Okay.
They're
I haven't seen
studying,
that one. I
they're
haven't seen that
studying
whale
the
fall.
zombie worms. They're very interested in zombie worms.
Oh, aren't we all? I have no idea what they are, but I'm super interested.
You can find links to that information on our website, nautiluslive.org. Just click on gallery. One of our main gallery options is the whale fall that Dan was talking about. You can see the first time it was discovered, and then it was revisited later to see how much of it had been consumed by zombie worms, I guess.
There was an event this summer in the Atlantic off of Cape Cod where there was a dead whale floating and a number of great white sharks feeding on it at the surface.
Oh, wow.
Then it made me wonder, how long do they float before they fall? If they always float first?
Are we nearing the end of a ship move?
We were. I just bumped another move up,
Okay, how much?
50 meters.
Right.
But I can come to an all stop when we need to.
A brittle star on the move.
Yeah, they move surprisingly fast. Oh, he's going for his cliff jumping, little base jumping brittle star.
Adrenaline junkie brittle star.
Yeah.
Some nice sponges up ahead.
Oh, yeah.
Sponges on the horizon.
So many.
A big one off to your left there.
Yeah, we're just about up back up on the ridge, spine of the ridge.
We'll be handing over to the next watch in just a few minutes. If you have more questions, get them in, and if we don't get to them on this watch, maybe our partners on the next watch will we'll get to those questions for you. Thanks so much for joining us here today.
Steve, can you get a zoom? I'm looking at the base of how that sponge attaches. It's pretty wild looking.
Yeah, very fibrous.
Yeah.
It is wild.
A scrub brush.
How much left on the move, Kate?
36 meters.
Let's
Five.
we'll stop at that one for shift change.
Yeah.
If you don't mind.
You want to come to a stop right now?
No,
at the
we'll
end.
let this move play out. Unless
Awesome.
back row
Thanks,
wants
Steve.
to stop sooner.
That's great.
No, I think we're good.
Yeah, I think it'll be okay. It'll still be a good place to hand off. Interesting place.
Yeah, Argus will still be swinging, but
Sorry, so that was not clear. Would you like me to come to an all stop?
no, we'll let this one play out,
Copy.
and then that should be it for this. For this watch. We don't have to stop during the shift change, but it's kind of nice because sometimes gets a little distracting.
Oops, sorry. Seeing these any ripples?
No.
Yeah, there's a few.
Oh, do you see
small
some?
ones,
Where do you
yeah.
see them?
Bottom of frame.
Bottom right.
Oh, bottom frame.
What are you looking at?
Ripples. Ripples in the sediment.
Oh, yeah, yeah.
You can see them in Argus view a little bit as well.
That's like a totally coreable there. That's got to be like at least a core deep.
About time to go pick through some Thanksgiving leftovers.
Yeah. Pumpkin pie for breakfast it is.
There was no leftover turkey.
There was
No.
no
Really?
leftover
So
turkey.
we did well.
Uh-huh, we did do well. That was good turkey, too.
Yeah,
It was.
moist.
Yeah.
Cranberries and oranges in the middle. And Mark got talking about trash can turkeys. out and buy a new trash can, turn it upside down, stick a can of beer up the turkey, and roast it.
So easy.
I'm sure it's delicious.
So gonna try that when I get home.
Zoom. See if I can hold it.
Call that beer can chicken.
I don't want to get too close to the bottom or thrust too hard because I'll end up with like tons of sediment.
See, I always, when we look at them, they always look a lot
You
like
don't
the
think that'll
soft coral,
whip
so
up?
we look like.
No.
Okay,
Not,
go wide.
Not if you land nicely.
Same texture.
Yeah, same texture.
The stuff on the rocks has been flying up. That's been pretty light.
There's a soft coral.
We're kind of headed east now.
Seems like that ship move is finished, huh?
It's still moving at 0.2 according to my graph on it.
Okay. There's some
Yeah,
nice
we've got 12 meters left in the move.
big sponges up there.
Oh, yeah.
I'm gonna zoom up to big sponge land there.
Okay.
Then you'll have to pass big corals. I know, difficult.
I don't really like passing big corals. I want to look
Me
at
neither.
them. But I also want to look at big sponge land, so
Chris will be happy.
yeah. Let's get them a big sponge.
Looks like you got your choice of both.
I know. Look at
Yeah,
this.
ooh, this is a
place.
nice spot.
Godzilla egg over here.
You're
We're back in-
welcome, 4 to 8.
Candyland.
And we're right back almost on the top of the ridge there.
Look at all these amazing critters.
I'll be curious to see where they head next. All right, nice watch, everyone. Video.
Yeah, great watch.
Switching over.
Yeah.
Thanks, Steve.
Thanks, Steve.
We came, we saw, we kicked sponge.
Well, I don't think that that's allowed in the permit, kicking sponges.
That's definitely not in the permit.
All right, friends. We got folks coming in. We got folks coming in to relieve their replacements.
It's another big sponge up the hill there, Gummy.
We're getting
But
great views
I like
here.
this sponge.
We hope that you stick with us. We have the rest of this
Run
dive
over, though.
and
Okay, fine.
several more dives coming up over the next week or so. You can always check our website,
The move is just
nautiluslive.org,
finishing, by the way.
where you can
Okay.
find out our upcoming dive plans. Wherever you are in the world, good afternoon, good morning, or good evening. And this is the Tectonic Turtle Watch. Checking out. We'll see you next time.
So our scientist ashore is saying the texture of the anemone and the soft corals are actually quite different. Even though they look similar, the surface texture of soft corals are not as soft as you imagine, more like sandpaper.
Yeah, they're not gelatinous like a... Looks like the ship's coming to a stop there. Morning. Okay, that's Dan signing off. Thanks, everyone.
Yeah, up on that wide ridge. I don't know. I don't know what's next. Guess there's been tracking issues off and on through the nights. The USBL was not ranging well, just all over the place. But it looks pretty good right now.
Good morning, Aphrodite.
Good morning, everyone.
Good
Morning.
Morning.
Good morning to all of our viewers. We are starting our watch from 4 to 8 a.m. Thank you for following us on nautiluslive.org. This is your Science Communication Fellow, Afrodita Fuentes. And so do you want to introduce each other? Or?
Should we do the ceremony first?
Oh,
or?
the ceremony? Yes, let's do that. Okay, as we start our watch and we come to the same mindset, we are going to conduct our private ceremony right now. So we're going to go right of silence to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. We will be back on SPL shortly.
Okay. I think I keep getting it wrong. It's Aphrodite, not Aphrodite. And I keep saying Aphrodite, don't I? Sorry.
It's okay. But I like Aphrodite better.
Okay, well, I will.
Because that's your name.
It's okay, his name's Crust, so.
But I'm a scientist, so we can pronounce. We have different pronunciations for the same thing, so.
That's right.
We can get away with it.
Well, I know what you're talking about, because I remember years ago when I was working overseas, somebody didn't know how to pronounce my first name, and they called me Christ.
Yeah.
Chris. So I was like, I don't think so.
Oh, you should have gone with it, Chris.
No, I wasn't going to go with that. But anyway, I'll get it right. So I'm Chris Kelly, and I'm sitting next to Aphrodite.
Yeah. All right, so what are we at?
What do you want to do?
What do we want to do?
You want to hear what they've been doing, or do you know?
I know, because I have been up quite a bit today.
Oh.
How about taking a look at that sponge right to the right, going off screen in the lower right corner?
I got it.
It
All right.
could be something different.
Something different.
So what we're going to do, since we're on the same basic site, is we're going to be looking for collectibles. The only thing I don't want to do is retrace our exact track, if that's possible. We could either go on one side or the other of it, or crisscross it, or do something. But I don't know if you can figure that out. But
Oh, I can make something up.
okay,
cool.
That'll work.
Can you come down, please?
It's kind of a barrel-shaped sponge I saw there. At least that's what I thought. Not sure where it is right now. It was an optical illusion.
Polyoptical.
Polyoptical. Never mind. Okay, it is 4 o'clock in the morning here.
Oh, yeah.
So we tend to hallucinate sometimes.
Nothing here interests you?
No, not really. So.
Okay, Trevor, so what do you think? Last watch was going down this slope and back up. We could keep playing that game if you want.
Nah.
Sure.
Was that a no from the yes, no? So yeah, you don't want me to make it up?
I saw what they were doing, and it basically the community tapers off at about 50 to 100 meters. And so I would just soon stay up on the crest where there's a higher density and more chances for encountering something new, if that's okay with the front row.
Yeah, it's fine. So do you want to come around this way?
Yeah, that would be good. Thank you.
All right, let's try that then.
Is that due south?
More or less. There's a lot of targets there, so they did kind of go through that area already, or we did on the last cruise.
Okay.
But I guess we can just scoot through that and then come around this way. Yeah?
Sure.
Okay.
Sure.
All right, then yeah, let's do some due south. It's a lot of south through stuff, but I guess it'll do. All right, how about 20 meters 180?
Raj.
Bridge nav. We get a 20 meter step 180. Thanks.
If we stay on the crest, I'm thinking we could stay a little bit closer to the bottom so we can see potential collectibles a little bit.
Yeah, we're going to be going maybe downhill a little bit. We'll see.
Okay.
So, Chris,
see how it goes.
To get down to this other side of the ridge, I guess, we do have to do quite a bit of south through stuff we've already seen, but I don't really know what else to do to get around there unless you want it to stay up on this side, if you could see my mouse moving really quickly.
No, let's go ahead with the plan. That's fine. That'll work out.
All right.
Thank you.
Yep.
As I mentioned to Aaron the other day, I have to be very careful with my odd sense of humor. I let out a big no the other day, and it was a joke, and it was almost taken seriously. So I will try not to do that. I just realized I did it again. I said, nah. So I won't do that. Okay.
Ship's moving.
Ship is moving. ROVs are moving.
Mind is moving.
The target is moving on this dive in general.
And as of now, we have about 2 hours on the bottom before we have to come up, just as an update on our status.
We are the blue water team, Chris. How can you change that?
We are blue water divers.
Please make it stop.
I have only one story I can think of today that might be of interest. The tank is running dry.
No, not possible.
Well,
I don't believe it.
You know, I've got a few more, but there's one that's potentially interesting, but we'll wait.
Sure,
and see.
bigger steps. Gonna ration it out. So we didn't finish our introductions. Aloha, I'm Andy Collins. I'm the education coordinator for NOAA's Office of National Marine Sanctuaries Papahanaumokuakea Marine National Monument. And to my right, I have Kainalu.
Yeah, aloha kakahayaka. I'm Kainalu, and I am the data logger.
And picture taken.
Yeah.
Look at that current.
There's a few people up front. You know, I can't tell who they are because I just see the back of their heads, but maybe they could tell us who they are.
We're the Mystery Science Theater.
Aaron Heffron, navigator.
Trevor Shepard, Herc.
Antonella will be Argus.
And Aaron Rainey, video engineer.
All right, morning. Yeah, so this is still on Don Quixote Seamount. This was particularly productive. I think we're relatively close to the area we're in now earlier, 2 days ago, before we had to run south to try to get away from some swell, which was
we're
very
really
successful.
close. We're in it.
We're in it. All right. Well, that's even clearer.
How do they always set this to useless mode?
I don't know if they don't want to actually see anything, I guess.
The reason we came back here, and I was explaining earlier on when we were descending to the seafloor for this dive, was that we wanted to drop in about 1 kilometer further down slope from the start point from the other day when we found this place.
Well, maybe
the idea was to try to see possibly how far down it goes, and where we started, to me, was a logical start point for this community because I was looking at the topography, and I was absolutely delighted to be wrong. There were fairly dense patches of this community that extended below our previous start point. So it's pretty interesting.
So what was the extent? What was the band? What's the band?
Well, we landed on a nodule bed, and we collected some nodules, and then we tried to do a sediment core sample, but the sediment is not very deep.
Yeah.
So it only went in a little bit, so that was abandoned. And then we were kind of off watch pretty quickly after that, and I was watching for a little while, and they were going up sort of a rocky little knoll there, and that had very close to this same type of density up there. And then they got some other highs when I woke up this morning, and explosion of biology all over the place. And this was before the start. So that's not it. They're not just restricted to these very tight ridges, which I thought they were. That doesn't seem to be as important.
But the depth was, so like 2,100 or so is petering out a little bit.
Not so much. We've seen communities like this all the way down to about 2,500.
Okay.
So are there any similarities between slime molds and sponges?
Slime molds. Boy, I'm trying to remember what those are. What are slime molds?
I don't know. Probably in the shower.
No, I think it's terrestrial, isn't it?
Yeah.
Yeah.
Slime mold.
You know, I just have forgotten. What's a slime mold?
Let's look it up.
I've heard of them.
Yes. Informal name given to several kinds of unrelated eukaryotic organism that can live freely as single cells, but can aggregate together to form multicellular reproductive structures.
And here's a bunch of pictures.
So yes, terrestrial.
Those are quite pretty, some of those actually. They've got an awful name.
Look, one on a petri dish too.
Yeah. Sponges are sort of an aggregation of single cell animals, and they do coordinate to form a cohesive structure, but it isn't just for reproduction. It's,
I'm staying a little.
It's, you know, they're
okay.
together.
Cool. Yeah, and then you're maybe not going to be directly on those points, and that'll be great.
So the sponges are going to be in a more advanced
Spongy.
form of that. Plus, these must be plants, right? Or I don't know what they are.
Well, it says
fungus.
it used to be in the fungus kingdom, but now it's moved into the kingdom Protista.
Protista, okay.
Which one used to be in the fungus kingdom?
Slime mold.
Mm.
Were formerly classified as fungi, but are no longer considered part of that kingdom. There you go, more taxonomy, switcheroonies.
Well, anyway, the viewer's partially right, I guess, because these sponges don't have, I mean, they have differentiated cells to perform different functions, and they could secrete different types of microscopic skeletal parts.
Bridge nav, another 50 meters, 180.
I like calling it the taxonomic switcheroony. That's a good technical term.
Wow, this is quite interesting distraction here. There's all sorts of people that are trying to teach slime molds how to navigate mazes.
Of course, that seems like a reasonable, normal hobby.
Absolutely. It looks like a best Christmas gift.
I beg your pardon.
The slime mold maze? Come on, under the tree?
Okay.
Every kid wants one.
Okay.
I think every kid wants one because it's gross, and kids like gross things.
It's true.
Not just kids, I kind of like them a little.
I know. I figured. That's why I mentioned it.
All right, we're coming down a little bit. Again, this is, I keep forgetting that this perhaps is a new batch of people who might be watching. This is one of these massive glass sponges, Poliopogon, interspersed between a variety of corals. Most of these unbranched corals are probably bamboo, unbranched bamboos maybe, I think. Some of these fan ones are in a family called Primnoidae, and also they're bamboos here as well. These kind of masses of brown and white in here are presumably the dead bases of massive sponges. So they were there on the seafloor, and then something occurred. They just died out, old age, or got blown over or something. And the holdfast, if you will, is still stuck to the bottom.
Chose the wrong rock.
Well,
that's
no,
my favorite.
These are okay, but it may have been they just died, or who knows? And who knows how long ago they died.
And is there a way to age the sponges to figure out their ages or any lifespan range?
People are trying, but right now that's sort of an evolving science, if you will. I don't know of any consistent way to do it, but it's probably through isotope analysis or something like that. They don't have any rings. They don't have any structure that allows you to easily determine their age.
I think I'm going to grab one of these pictures of the sponge that attached to a rock that was slightly too small for it to hold in the current, and it fell over on its side, you know. I think I'm going to make a demotivational poster out of that. Bad life choices.
Could that be an anemone right there, the reddish, pinkish part?
Anemone.
Oh, that's slow. Yeah, let's go faster. You said someone said they wanted to zoom on the anemone. Let's go for it.
Sure.
I think that was
Go ahead.
our
That's a nice
videographer, Aaron.
Take a
You want
picture.
to get a picture of this?
That wasn't me.
Oh, somebody else. Okay, sorry.
Could have been anybody.
It was me. I like anemones because they're red, like my hair.
Hair's not
relate.
quite that red, I don't think. But anyway,
It was.
This anemone is most likely in the family Actinostolidae. And most of these anemones we can only go to family because a lot of their taxonomy is based on internal parts that we can't see. Could we look at this fan if we're right here?
Yeah.
Just a close up on it.
Go ahead and zoom.
Beautiful.
Yep.
Looks quite complete.
I have more zoom if you want it.
Yeah, if we could, please. Okay, so this is not a bamboo coral. This is in the family Primnoidae. It's a beautiful example. My guess would be it's in the genus Paracalyptrophora. There's a couple unbranched bamboos right now that are in view.
Hey, Chris, are we going to get to do a cliff dive on any of this cruise? Or is it all going to be ridge tops?
The next dive, it's a ridge, but it's a very steep ridge. So I think that qualifies as a sort of a cliff dive.
All right.
Do you like rock climbing, just out of curiosity?
Sorry, what's that?
Do you like rock climbing, just out of curiosity? Okay, well, that's where that's coming from, I guess. Me, I like couches, TVs.
I think we finished exploring all the couches down here already.
Oh. Spoken like a true sponger, Chris.
This sponge looks really comfy.
Maybe that's why I like sponges. They kind of resemble couches, right?
Chris has a sponge-shaped couch at home, I've heard.
No, that is not true.
He has a couch-shaped sponge.
But which species would it be? Which would be the most comfortable?
The lazy boy sponge.
Lazy boy sponge. Some of these actually look like they've retracted too. I don't know if it's the current that kind of caused them to bend over backwards the way they grew.
One of our viewers noticed the coral had a very square base. That fan.
A very
that
what?
Fan.
Oh, very square base. Oh, okay.
Those large fan-shaped.
So I saw a sponge on this site the other day that I didn't know. It was sort of brand new, and I didn't want to get too greedy. We already had 2 sponges in the box. But I've got my eye out for this. Hopefully, I only saw one the whole dive, so I'm hoping we can.
What should we be looking for?
It kind of looks, it's pretty big like these, but then instead of sort of sticking up sideways or on a stalk, it was flat, looking straight up with some big holes. Didn't have sieves. It looked like a turbo fan facing up or something, I guess.
So you have these, are there only 2 different attachment approaches
50 meters,
for sponges?
180.
The cementing and the fiber, what's it called, fiber attachment?
So the fiber attachment, the formal name, I'm sure everybody is really fascinated to hear this, is lophophytus, and that's what they call it. And when they glue themselves with silica onto the substrate, that's called basophytus.
Lophophytus and basophytus.
Yeah. And if you think about a sponge and what it is, that's pretty much all they can do there, right? They can either just put down bare spicules and grapple the bottom, or they can just secrete a lot more silica and glue themselves to the bottom. There's not much else they can do.
Yeah.
So Aaron does have us going on a track. We are not repeating the track that we did that first day.
Well, we kind of just were for a little bit, but we're almost through that.
Okay.
We're soon on fresh territory.
Okay. Wow, from the Argus view, you can really see the current, huh?
No current's important.
Is that big one a Poliopogon as well?
Yep.
Right.
Yep. All of these are different varieties of Poliopogon, and a lot of them are the same species. They just have different growth forms depending on how they're interacting with the current and their substrate. That's what's so tricky about sponges. You know,
that's a pretty big one coming up there. I wonder how many organisms are living in it.
Go ahead and zoom.
It almost looks like one that we saw last dive too, that's got the double faces.
Uh-huh.
But I mean, there's going to be so many of these. Hard to say if this particular one is something we came across last time. This is Poliopogon.
Yeah, it has two concave faces.
Yeah, so some of them only have one. That's part of their taxonomic distinction. Some of them have that little stalk. You see a lot of those kind of sticking up, and these don't.
There's a little bathypathes in there, black coral on the shade of that.
You know,
we're going this way.
Asako likes to hit a man when he's down.
I
he
saw
goes,
that.
High blue water lead.
I saw that. We are not. We are hui huakai, team sponge.
I like that. Good poke there. Good tease.
Is that the sponge that you're looking for?
No, that's one Emil was interested in asking me about. That one there. That's one that's fairly well known. No, if we see that sponge, you'll probably see me jump out of my seat and yell Eureka or something.
Which sponge is that?
It's just one that I've never seen before. I kind of like to see ones that are just they're very different. This looks like, I wonder, no, I guess we're just up high. I wonder if we should now start turning a little bit toward the east a little bit more.
So you want to start swinging it around now?
Yeah, it's starting to taper out a little bit. And that's what happens.
Well, Trevor's just looking over the edge. It's not really a reflection of everything, but I could start bringing it to, let's see, like 147, 148.
Yeah, just kind of like a parallel track, but staying on the crest if you can.
Okay.
Or the couch, I mean.
The couch.
Sponge couch.
All right, how about we 145 it
Sponge
then?
lounge.
145. Oh, okay.
Bridge nav?
What is the current, Trevor? Do you
get a 50 meter
What
step?
is the current?
145.
It's the flow of water.
I know, thank you. Oh my gosh.
Are you asking me direction or magnitude?
Yeah.
Fair enough. It's plenty, and it's going from right now left to right. It changed a lot. I just came down the side of that ridge, and the direction might be different, and I don't remember what it was, so that's not very useful either. Yeah, I'm kind of in a little nook here. Like when I get back on top of the ridge, ask me again, and I'll tell you.
Okay.
But it's kind of getting some weird swirling here. I don't know.
Yeah, it seems to be going in multiple directions, but there seems to be quite a fair amount of content in the water column.
Yeah,
different. Totally. It was clearer the last time we were kind of coming through this area.
I think it's heading west. Or it was in the top of the ridge anyway.
About a knot or two? I mean, that's kind of...
not quite that much. Probably, let me think about that. Probably half.
Okay.
That's totally...
How do you guess that?
I'm guessing that based on how much lateral I can use. I cannot lateral a knot. So if it was a knot, I'd be falling off.
Okay.
And I was probably, to stay in position, probably 50% lateral, which would be about half a knot. I don't know.
Sounds reasonable.
Very imprecise measurements. What are we doing, 145?
145, yeah.
Okay. Seems like no matter what we pick right now, we're heading down slope. It's okay, probably just a little nugget.
Just a little divot.
Yeah. I see the next one coming up.